Our first presentation from the LTRs is Deron Burkepile from the University of California, Santa Barbara. He's from the Moorea Coral Reef LTR, which of course is in French Polynesia. The award for this LTR is at UC Santa Barbara. You're a researcher associated with the LTR, and Moorea Coral Reef LTR has been in business since 2004. So it's a little bit of history there. So Deron, I'll leave it with you.
Oh, thank you, Dave. Morning, everyone. I'm excited to be here today to talk a little bit about coral reef ecology. And when most people think about coral reefs, they get a picture in their mind that looks something like this. Lots of really pretty aquarium fishes swimming around, lots of pink and purple corals, right? Lots of three-dimensional complexity. But what people often don't realize is that this ecosystem, the engine that runs this ecosystem, is a mutualism between the coral animal and a little single-celled algae called Symbiodinium that lives within that coral. Right, so that zoomed-in picture there is a coral polyp, right? One of the individual animals, and that kind of golden shading in there is that single-celled algae that lives inside that coral's tissues. So this is a classic mutualism, right? The coral gives those little algae a place to live, gives them nitrogen and phosphorus that those algae need for photosynthesis, and in return, those algae give the coral the carbon that it fixes from photosynthesis. And this mutualism is the engine that creates these giant biogenic three-dimensional structures that are one of the most diverse ecosystems on the planet, but are also really important. They're the engines for tropical economies all over the world, from tourism and fisheries. They generate billions of dollars every year and feed about 1 billion people on the planet. But if you've been paying attention to the news at all recently, you know that coral reefs are in trouble from a variety of global and local stressors: climate change, ocean acidification, overfishing, nutrient pollution. And this was really reinforced over the past couple of years, where we saw worldwide bleaching events, where increases in thermal stress causes that coral-algal partnership to dissociate, corals bleach and often die. So on the Great Barrier Reef alone, between 2015 and 2017, we lost probably between about a third and half of the corals that live on the Great Barrier Reef from these coral bleaching events in response to climate change. And the fact that these coral bleaching events are coming more frequently, and they're getting bigger and more intense, and you've got other stressors like overfishing and nutrient pollution, some people have led that to predict that coral reefs will disappear by the end of this century. That they'll turn from reefs that we see today that have lots of really big fishes and pretty corals, and they'll turn into essentially zombie reefs with very few fishes, very few corals, and are covered in these really harmful seaweeds. But I have to say that I'm an ocean optimist. I really think that we'll have coral reefs in perpetuity. And one of the reasons I think that is that these healthy ecosystems are really benefited by fishes. So today I'm going to tell you why good fishes make good reefs. And I'm going to give you 3 take-homes, right? So the first is that fishes are really important herbivores. They eat seaweeds, that facilitates corals, and it especially helps reefs recover after disturbances like coral bleaching events that kill corals. Second, that fishes are important sources of nutrients that actually help corals grow and can help corals withstand these harmful thermal stress events. And third, all is not lost. Local management decisions that we'll talk about can actually help corals and coral reefs withstand the Anthropocene. Okay, so to tell you all this, I'm going to take you all on a little field trip. I'm going to take you out to Moorea, which is almost literally in the middle of the Pacific Ocean. You can center the globe on Moorea and basically see no continents, which is pretty cool. All right, so this is Moorea. It's actually where several movies have been shot. It's the model for Bali High. I think Mutiny on the Bounty was shot here. That's Tahiti off in the background, so we're kind of looking west to east across Moorea to Tahiti. It's a beautiful place to work. It's tough to convince my wife and kids that I'm going to work when I go to Moorea for 3 weeks, right? We do spend 10 hours a day scuba diving all day long, but we're doing it in places that tourists spend thousands of dollars to go on vacation. It's not exactly a hardship, right? So the reason we work in Moorea is because that's where the Moorea Coral Reef LTR is. That started in 2004, as Dave mentioned. And our home is the University of California Gump Research Station that's run currently by UC Berkeley. And the Gump Station has been really well supported, especially by Gordon and Betty Moore and some gifts from their foundation since the early 1980s. And they've been really generous to the Moorea LTR as well. The Gump Station is located on the North Shore, that little red dot right there, at the mouth of Cook's Bay, which is named after Captain Cook, the famous explorer that spent a lot of time in the world. There are 19 associate investigators spread across many different universities. And if we kind of zoom into the north shore of Moorea here, we can see the three different types of coral reefs that we have on the island. So starting right next to land there, we've got the fringing reef, which, as it is aptly named, grows right on the fringe of the island. You can basically jump in from the beach and be swimming around big corals and coral heads with lots of fishes. And then if we start swimming offshore, we get to the lagoon area, these back reef areas that are kind of this matrix of big coral bommies and sand and about 10 to 12 feet of water. If we keep swimming offshore here, we'll come to the reef crest, where the waves actually break at the surface right on the reef. If we swim over the reef crest on a nice gentle day, we can actually then swim out onto the forereef, which slopes from right at the water's edge down to hundreds of feet over a pretty short distance. And these three different types of reefs have different species of fishes, different corals, lots of different topography on them, and they're also really differentially affected by different human stressors, things like fishing and nutrient pollution. And if I were to tell you that one of the biggest threats to these reefs, especially the forereef, is a starfish, you'd probably say, that's ridiculous. Because starfishes are cute, they're adorable, they're pretty, but I'm not talking about that starfish. I'm talking about that starfish, right? That is the crown of thorns sea star. It's about the size of an extra large pizza. Those are pretty nasty spines that can give you a toxic poke and give you a pretty nasty infection. And it's also basically a coral murderer, right? It's a coralivorous sea star. It loves to eat coral, right? So it climbs up on a coral head, it everts its stomach, and dissolves, digests all of the coral tissue off of that live coral. Once it's done with all the tissue, it climbs off, and you have this bright white dead coral skeleton behind, right? And the unfortunate thing about these crown of thorns sea stars, they're also called COTS, as an acronym, is that they form these big outbreaks, especially in places where their predators, big fishes, have been overfished. And they form these huge waves that literally eat their way across a reef. And they've been one of the biggest sources of disturbances to Moorea. They get outbreaks about every decade or so. So when the Moorea Coral Reefs LTR started in 2004, this is what the forereef looked like. Remember, it's that gently sloping reef out on the very farthest reef away from the island. Had about 50% coral cover on it. Then 3 years after the LTR started, a big outbreak of these crown of thorns sea stars happened, and we were like, oh no, it's going to eat our field site. And it literally did eat basically all the corals off of the forereef before these starfish eat themselves out of house and home and then starve and the outbreak dies back. And then to add insult to injury, less than a year after the COTS outbreak started to fall apart, Hurricane or Cyclone Ollie basically ran right over Moorea. So the crown of thorns starfish ate all the corals, Hurricane Ollie destroyed what was left, and in 2010, the forereefs looked like that, right? Almost literally all of the corals were gone, down to 2% coral cover. But these reefs are really dynamic ecosystems. This happens naturally all the time, so these ecosystems started to bounce back. And 4 years later, in 2014, we had 30% coral cover. Little coral larvae recruit out of the water column down to the benthos. They start growing into bigger corals. And just this past year, our data showed that these reefs have basically bounced back to even higher coral cover than it was before the outbreak, right? 70% coral cover. So in less than a decade, the corals on these reefs have basically bounced back to even higher abundance than they were before. And one of the main drivers of this really rapid recovery is the abundance of herbivorous fishes, like these parrotfishes here on the top and surgeonfishes here on the bottom, right? And these parrotfishes are really aptly named. All of their teeth are fused into a beak-like mouth, right? And you can actually literally hear them eating on the reef. You can hear the parrotfishes bite as they scrape the seaweeds off the surface of the coral matrix. These surgeonfishes also really important herbivores. They're called surgeonfishes because they have these really sharp scalpels on the base of their tail. They use it to fight their friends and ward off predators. And anyone who's been dumb enough to catch one of these with their bare hands, I'll admit to that, finds out very quickly how sharp those scalpels actually are. And you can think of these parrotfishes and surgeonfishes essentially as marine goats. They swim around eating seaweeds all day long and basically keep the reefs clean. When you have these abundant fishes, these reefs are relatively free of big seaweeds, and they keep coral reefs healthy, allowing adult corals to grow and little baby corals to settle and recover. Right? So these are the dynamics that we've been seeing in the Moorea Coral Reef LTR. Right? You had these big outbreak of crown of thorn sea stars, 2006 or 7 to 9. They basically eat themselves out of house and home. That outbreak collapses, and they've been pretty much absent ever since. Right? They ate corals basically down to nothing, but we see this huge recovery in relatively short order. And the driver of that, as I said, was that the herbivorous fishes kept these really harmful macroalgae at low abundance and allow these reefs to stay in a state with these little bitty filamentous turf algae that signifies a healthy reef and allowed corals to recover. And one of the interesting dynamics here is we saw a really big increase in the abundance of herbivorous fishes after all these corals died. Right? You had more space that was colonized by seaweeds, more food for these fishes to come in and eat, and we saw a really big population explosion of these herbivores. They came in, they basically absorbed all that extra production from the seaweeds, and allowed that really fast recovery of corals. So this coral recolonization requires the low cover of macroalgae, and that's driven by the abundance of these herbivorous fishes. But there's also a really cool connection between the different reefs in Moorea that allowed this response to the herbivores. So if you jump into the fringing reef right along the island, you'll see corals like this, right? So this is a big Parrotfishes rust coral. It's probably bigger than I am tall. It's probably 10 feet wide. And these is all across the forereef, and they're a really favorite habitat for these adorable little baby parrotfishes. Right? So these parrotfishes settle as larvae out of the water column to the benthos. They like these big Parrotfishes corals. And then over time, they essentially migrate out to the forereef where they become adult parrotfishes. Right? So it's this connection between the lagoon reef, the fringing reefs, and the forereef that allowed this big population explosion of parrotfishes that absorbed all those seaweeds and helped these reefs and corals on the forereef recover. So that's the story of the forereef in Moorea, but if we go to some of these different reefs, we get a different picture of how the dynamics of reefs work. So if we go from the forereef out here to the left of the reef crest and swim back over and back into the lagoon, depending on where we go in the lagoon, we get a different picture of what these reefs actually look like. Right? So you can go to some areas where our long-term data show that corals have been really abundant throughout the course of the Moorea LTR, and macroalgae have been really low. But you can go to other areas in the lagoon, and you can see that corals are on this really precipitous decline, and macroalgae are on the rise. Right? These are big brown seaweeds called turbinaria that tend to take over the back reef when there's relatively low herbivory. And one of the drivers of these transitions is that there's a big lagoonal fishery. Right? So these are called tuis. These are racks of fishes that the fishermen set up along the roadside in Moorea. So Saturday morning, you can drive the ring road that rings the island. You can go pick out a nice big parrotfish or a nice big surgeonfish to cook up at a family dinner. If it's just a regular weekend, maybe you'll pick a couple of smaller fishes. Right? So there's a big fishery on these really important herbivores. And that's been one of the triggers, one of the mechanisms that's driven this rise of seaweeds in the lagoon, is removing the important top-down force on those seaweeds. But there's also another interesting thing that's going on in the lagoon, and we can track it with this seaweed. So this turbinaria, we can collect it from different places around the island, measure the nutrients in its tissues, and we can build an idea of where the nutrients are really abundant all around the island. Right? So we collect this turbinaria at about 200 places all around the island, and we can build this heat map of nutrient availability. Right? So warmer colors means more nutrients in the water, and you can see that the North Shore is really kind of a big nutrient hotspot around the island, probably because you've got these two big watersheds here that drain into these big bays, and it drains lots of pineapple plantations and, unfortunately, pig farms. Right? And we also see the rise of macroalgae in these places where we have these big nutrient hotspots. But in areas that are seeing less nutrients from the land, that's where we see corals persisting. Right? So you've got fewer herbivores to eat the seaweeds, you've got more nutrients to make them grow faster, and you get this transition to seaweed-dominated systems in some parts of the lagoon. But these nutrients have some more insidious direct effects on the corals, right? So we've got our nutrient heat map showing that the North Shore is especially enriched in nutrients. If we do these same surveys with coral bleaching, what we see is we see bleaching hotspots in those same places that we see elevated nutrients. Right? More nutrients out on the reef actually lowers the temperature threshold for these corals, makes them bleach sooner, even at lower temperatures, and leads to more mortality right? So we're facilitating the rise of seaweeds. We're actually literally killing the corals with extra nutrients, driving corals down, driving seaweeds up. And we've explored this experimentally. We've actually done experiments to look at how these nutrients and different kinds of nutrients facilitates coral bleaching. So we can go out to the forereef, which is naturally low in nutrients. It doesn't have that same kind of human influence from the land. And we can actually do an experiment. So we can swim out to the forereef and put out these nutrient diffusers, which is just basically a PVC pipe that we pack with nitrogen, either nitrate nitrogen or urea nitrogen, and look at the effects on coral bleaching, right? Those are two really unhappy corals next to a nutrient diffuser that's pumping out lots of nitrate. So what we've seen is that when we add extra nitrate around these corals, we see a big increase in coral bleaching, right? So the nitrate there is in the blue, the urea addition is there in the red, and what we see, even in the cooler parts of the years, we're seeing more coral bleaching when we add in this nitrate source of nitrogen, but really no effect of this urea source of nitrogen, right? So you're getting differential effects of these two different nutrient sources. And this increase in bleaching actually leads to increased mortality. So our common coral here is Spatial, it's basically everywhere. And when we add in nitrate, we see more coral bleaching, and we see more coral mortality. We kill about 10% of the corals by adding a nitrate, but really no effect of this urea. And I should note that these two sources of nitrogen come from different places. So nitrate is often a signal of human-derived nutrients from agriculture, from livestock, right? So it's something that humans are putting there that typically isn't there in very high abundance. Urea is a fish-derived source of nitrogen, right? So fish are recycling these nutrients on the reef, and we know that that's important. And anybody that's had small children or been around small children knows that everyone poops and everyone pees. And we know this thanks to this adorable book from Heterogeneity, right? So we can think about these fishes like this parrotfish that swims around the reef on Moorea. It's eating algae and maybe a little coral mixed in there. And then it's recycling nitrogen and phosphorus and a particular organic matter back to the reefs. And work from the LTER and other groups have shown that these fishes are really important for corals. So a lot of the corals out there on this reef have these little damselfishes and other fishes living inside and on them. And if you look at corals with fishes versus without fishes, the corals that have these fish schools associated with them, they grow faster and are generally overall healthier than corals that don't have these fishes. Right? So fish recycling these nutrients gives the algae extra nutrients for photosynthesis, it gives the coral particular organic matter to eat, and makes them healthier. So this led us to think, can these fishes, the association with these fishes, actually help these corals survive under thermal stress that would normally cause them to bleach? So we did a really simple experiment. We went out to the forereef and collected a bunch of this branching coral Spatial that had these adorable little damselfishes associated with it. We took these corals back to the lab, left half of these corals with their fishes, and took fishes away from the other half. And then we challenged them with temperature. We left half the corals at ambient temperatures, and the other half of the corals we started to turn up the thermostat and heated these corals to a temperature where they would normally bleach and potentially die. So what did we see? So first, at ambient temperatures, when we took the fishes away, right, the filled circles are corals with fish, the open circles are corals without. We take the fish away, and the corals grow a little more slowly. Not surprising, we expected to see that. When we take the fishes away and crank up the temperature, these corals get really unhappy, right? They basically didn't grow at all when you heat them and take away their important fish associates. But when you heat them and leave the fishes intact, they actually grew as fast or faster than corals with fishes at ambient temperature, right? Suggesting that these fishes are really important for these corals to withstand thermal stress events. Also suggests that if we limit the amount of human-derived nutrients that are going out to the reef, we can maybe protect corals from coral bleaching, lower their bleaching thresholds. If we protect fish communities, it suggests that their recycled nutrients can actually help these corals withstand really stressful periods on the reef. Okay, one more caveat about the importance of fishes before I go. Now, if I told y'all that this coral was in mortal danger, you might not believe me, right? It looks really healthy, there's no coral bleaching, there's no disease, but those are snails. I know it doesn't look like it, they're covered in seaweed, they're also they always look kind of snotty looking. But if you pull one off, it looks like this. This is a Coralophilia snail, and it's basically a vampire. So they're all feeding right on the margin of the coral colony, on the coral tissue right there. And they're basically sucking the resources out of this coral. They're feeding right on the coral margin, and that coral is mobilizing its resources from other parts of the colony, and these snails are taking it up. Right? So this coral, even though it looks pretty healthy, actually has relatively low energy reserves because these snail vampires are basically eating it away. So we were really interested in the role of these snails in coral health. So we decided to do an experiment. We found a bunch of these coral colonies that had snails on them. A third of them we left with the average density of snails. In another third of them, we removed all the snails. And then in the last third of them, we doubled the snails. We added extra snails. We just happened to do this experiment in a really warm summer, right? So the water temperatures heated up, these corals got stressed, and a bunch of them bleached. And they bleached basically like this. The corals with no snails bleached a little bit, but in general, they looked to be okay. The corals with average snail densities bleached quite a bit, and the corals with a high density of snails, almost all of them bleached 100%. Right? That coral's alive, but really unhappy. So then the water temperatures cooled down at the end of the summer, and then we decided to see how these corals responded to the bleaching event in snails. Right? Not surprisingly, the corals without snails were fine and happy. The corals that had average density of snails, a lot of those colonies lost tissue, about half of their tissue. So those corals died back about 50%. But a lot of the corals that had high densities of snails that bleached really intensely died. Right? So this suggests that these coral-eating snails are sensitizing these corals to thermal stress. Make them bleach more intensely when those snails are basically sapping up their resources that these corals use to deal with being bleached, and actually leads to increased bleaching and increased mortality. That's pretty depressing. But there is some hope, because when you go around to these reefs and survey the snail predators, like these big grunts and big lobsters, you see these relationships. With the more of these coral-eating species you have, the fewer snails you actually see on the reef. So you can go to a marine protected area there on the left, and you see really low abundance of these coral-eating snails. You go to an unprotected area there on the right that has lots of fishing, where these big coral-eating fish, snail-eating fishes and lobsters aren't very abundant, and you see lots of snails. Right, so this suggests that these marine protected areas, right, by protecting these corals from these additional stressors, could potentially help corals deal with the stress of climate change. Okay, so what I've told you today. Good fishes make good coral reefs because they're really important herbivores. They help keep reefs clean after big disturbances that kill corals, allow baby corals to settle, recover, and turn into big corals. Right? They're important sources of nutrients. They help corals grow, help them withstand thermal stress. They're important predators on little corallivorous invertebrates and help reduce the stress of corals and allow them to deal with climate stress. And local management decisions may be able to help corals survive the Anthropocene. If we can protect fish communities, lower the amount of human-derived nutrients that are going onto these reefs, we're maintaining these positive feedbacks that help reefs recover and maybe help them recover even in the face of coral mortality from bleaching and other stressors. And this gives me a lot of hope, right? Even though we see these big coral bleaching events, because these local management actions, if we can actually implement them, as well as be pretty aggressive in slowing our greenhouse gas emissions to help minimize the warming that our planet sees, these local management actions might actually be able to get us through the next 50 to 100 years, and then hopefully we'll be able to keep these. Thanks very much.
As promised, we have an opportunity to ask Dr. Burkepile a couple of questions if anyone wants to approach the microphone.
Yeah, fantastic talk. Question about that very important aspect regarding the recovery. So you have a cause event, everything dies off. And now I have a question regarding the chicken and the egg. Because in order to have the fish present, there has to be the habitat. So it seems like there's this very critical moment there where you have to foster coral recruitment to to establish enough of a habitat to get fish in. So could you speak to a little bit about what the conditions are that perhaps around Moorea promoted that recruitment moment faster than others?
Yeah, so that's a great question, right? One of the things that these fishes need to persist is the habitat that these corals create. And when we see these big bleaching events happen, one of the things that does happen to the fish communities is you lose a lot of the coral-associated fishes if the corals don't recover very quickly. And one of the reasons we saw this really rapid recovery of the corals and this massive recruitment event is, one, there's actually connections of corals between the lagoon and the fore reef. So some of the corals, the coral larvae were actually coming from the lagoon. Some of them were coming from other parts of French Polynesia, right? So not every place in French Polynesia, not even Tahiti, had that big outbreak of crown of thorns, right? So we were getting lots of larvae coming in from the nearby islands, right? So this local connectivity, both within the island and among islands, really facilitated that rapid recovery of corals that allowed these reefs to maintain the fish populations.
Maybe one more question. Anyone has one? And I wanted to remind, some people come in late. We ask the speakers, when you ask a question, if you please identify yourself and where you're from, if you could do that.
Okay, thank you.
And Devin will be around. Oh, I'm sorry. Let's go.
Yeah, Darren, that was a beautiful talk. I really enjoyed it. So as a microbial ecologist on these reefs, I tend to think about things from an equilibrium perspective between your host, your symbiodinium, and your microbiome, right? So I'm really interested in a little bit of what you were saying about how nitrates are potentially facilitating bleaching events, and we've seen a little bit of that from the idea of increasing temperature also increases diazotrophy, or nitrogen fixing in the coral, which is then potentially releasing the symbiodinium from their nitrogen-limited state, right? Which is just sort of throwing the equilibrium off. So can you speak to the potential mechanisms by which the urea is stabilizing that equilibrium, maybe from the holobiome perspective?
Sure. So I mean, there are a couple different things going on with the different nutrient sources. So for the symbiodinium, the algae that live inside the coral, to use that nitrate, they have to expend energy, right? So they've got that really reduced form of nitrogen. They have to use energy to convert it to a form that they can put into amino acids, right? So that's energy that's not potentially going back to the coral. So when we're giving them urea or ammonium that also comes from fishes, those are in forms that are much easier to use for the symbiodinium. And there's some great data from our postdoc that shows that when you give corals ammonium, they actually get more carbon back from the symbiodinium. But if you give them nitrate, they actually get much less carbon back from the symbiodinium. Right? So the coral, or the symbiodinium, is basically paying its rent at a higher rate when you're giving it these fish-derived nutrients. And it's basically kind of skipping out on rent when you're giving it this nitrate, right? So, I mean, you're decoupling this kind of trade-off between the corals giving nitrogen to the symbiodinium and these algae giving carbon back to the coral when you're giving it these different forms of nitrogen. Okay. Thanks.